Okay, let's unpack this. We are diving deep today into one of the most exciting and, frankly, most demanding fields in modern medicine, precision immunotherapy.
Right. This is the ultimate promise of tailored cancer treatment.
But it's a promise that is currently hitting these massive, really expensive roadblocks. Our subject today is how artificial intelligence, specifically a monumental new multimodal model, is creating a kind of parallel digital universe of cancer patients to blow past those barriers and accelerate discovery.
That's the perfect framing. I mean, precision immunotherapy holds this immense, almost life-altering promise.
Right.
The idea of moving beyond one-size-fits-all chemo to tailoring treatment, not just to the cancer type, but to the unique, intricate way that your body and your immune system are interacting with that disease.
But scaling that knowledge has been, well, nearly impossible.
Exactly.
And that difficulty is precisely our mission today. We're examining a revolutionary solution called GigaTIME. It was developed through this really powerful collaboration involving Microsoft Research, Providence, and the University of Washington.
A real powerhouse team.
And they're promising to overcome these chronic data and cost roadblocks by generating this massive, unprecedented pool of virtual patients, all derived from routine data.
You know, the core insight here, which is truly driving the acceleration of precision health, is understanding that the digital transformation that's already happening in healthcare, coupled with the sheer power of the generative AI revolution, lets us extract knowledge from cheap sources that was, previously reserved for these expensive high-tech labs.
So you're saying we can find the secrets in the data we already have?
We can. We need to decipher the grammar of the tumor microenvironment, the time. But historically, getting the high-res data to understand that grammar at scale was cost prohibitive.
And the source material detailing this massive breakthrough is research published just recently on December 9th, 2025, in Cell. This paper describes how GigaTIME performs a kind of translational magic.
It really is like magic.
It takes those cheap, routinely collected pathology slides, the ones every cancer patient generates, and turns them into incredibly complex, high-resolution spatial proteomics data, virtually.
It's not just a shortcut to knowledge.
No. It's the democratization of the gold standard of data. So let's begin at the beginning. Why is the tumor microenvironment, or TIME, so absolutely critical in modern oncology, and specifically in immunotherapy? Why can't we just look at the cancer cells themselves?
That's a great question. The TIME is so much more than just the tumor cells. You should think of it as the entire ecosystem that surrounds the malignancy.
The whole neighborhood.
The whole neighborhood.
Right.
It's this dynamic, evolving landscape made up of immune cells, fibroblasts, blood vessels, the extracellular matrix, everything. And understanding this entire environment is fundamental because the TIME, it dictates the battle. It's the crucial determinant of whether a patient will respond favorably to immunotherapies like checkpoint inhibitors.
And this is what dictates whether a tumor is classified as hot or cold.
Right, precisely. A hot tumor is one that has successfully recruited or been infiltrated by the patient's immune cell, T cells, macrophages, and others.
So the immune system is already on the scene.
It's already engaged. And if it is, current immunotherapies are much more likely to work because they just need to be given the go signal.
And the cold tumor.
The cold tumor, by contrast, has found these really sophisticated ways to exclude or suppress or just hide from the immune response.
I see.
So if we can map out the components of the TIME in this granular spatial detail, we gain two essential things. First, the predictive power to tell us who will benefit from existing treatments. And second, the actionable insights we need to inform strategies to try and, reprogram that cold tumor to heat it up, making it visible and vulnerable to treatment.
That sounds like the holy grail of immunotherapy research.
It really is.
So to truly map that intricate cellular architecture, not just what cells are there, but where they are relative to each other and what state they're in. Researchers need the absolute gold standard in imaging.
They do, and that gold standard is multiplex immunofluorescence, or mIF. Yeah, mIF technology is incredibly powerful. It allows researchers to stain a single tissue sample for multiple protein markers at the same time.
Okay.
This means they can assess the internal states of individual cells. Are they activated? Are they dying? Along with their exact spatial coordinates within the tissue sample.
So it's the where that matters as much as the what.
The where is everything. That spatial data is the key to understanding the complex interactions, the key to deciphering that tumor immune system grammar.
And this is where we run straight into the bottleneck.
Yeah.
The economic and logistical one. mIF is the best data available, but the cost and scalability are just crippling the pace of discovery.
It is truly prohibitive. We aren't talking about a few $100s.
No.
Even for a single small tissue sample, just generating IF data that covers a couple dozen key protein channels, which you need to capture that system-level interaction, can run into the thousands of dollars.
Yeah,
often, between $3,000 and $5,000 per slide, depending on the panel size.
Thousands of dollars for one small piece of tissue. So if you, as a listener, imagine a large health system like Providence or any major cancer center, they have archives with hundreds of thousands, sometimes millions, of tissue samples. The physical remnants of patient journeys spanning decades.
Right.
That cost model means that even the most well-funded research teams can only touch a minuscule fraction of their available data. The wealth of potential knowledge remains locked away, inaccessible simply because of the budget.
That is the tragic definition of a bottleneck in clinical research.
Mm-hmm.
The data that could revolutionize precision oncology is just too expensive and too difficult to generate at the population scale that you need for major biomedical discovery.
And you need that scale.
Oh, you absolutely do. You need tens of thousands of samples to find subtle patterns across cancer subtypes and genetic markers. mIF lets you analyze tens, maybe hundreds, in a focused study. The scale mismatch is just vast, and this challenge has fundamentally limited our ability to conduct population-scale studies of the time. Until now.
So we have this critically important, high-resolution, super expensive spatial data EMF trapped on one side.
Right.
And on the other side, we have the ubiquitous, routinely available, cheap data that every clinic in every corner of the world produces. This contrast is why GigaTIME enters the picture as the great multimodal AI translator.
Exactly. We contrast mIF with hematoxylin and eosin H&E pathology slides.
Yeah.
If you ever had a biopsy for any kind of growth, you have an H&E slide associated with it. They are the absolute workhorses of cancer care. They're cheap, easy to process digitally into whole slide images, and they are routinely generated by pathologists worldwide.
And when we talk about the cost difference, it's just astronomical. The source material says these slides cost only about $5 to $10 per image to process and digitize.
A truly negligible operating cost compared to the thousands for MAMIFO. H&E slides are stained to reveal crucial morphological details of the cells.
What we can see.
Exactly what we can see. The blue stain shows the nucleus. The pink stain highlights the cytoplasm and extracellular matrix. And critically, pathologists and researchers know that this H&E-based cell morphology contains subtle, hidden information about the underlying cellular states and functions, even if we can't detect it with the naked eye.
Now, you mentioned that prior AI models have tackled H&E slides.
Yeah.
Just last year, we saw GigaPath, a foundational model that proved AI could indeed pull useful info from these huge gigapixel slides. What's the evolution here?
GigaPath was a phenomenal achievement. It demonstrated the power of scaling transformer architectures to interpret these enormous images. It showed it could reliably predict certain key clinical biomarkers, which is great. But those models were limited. They generally predicted only the average biomarker status across the entire tissue area. They couldn't tell you, for example, which specific cells were expressing a protein and where they were located relative to others.
So GigaTIME is taking that next huge leap by introducing spatial awareness.
It is. GigaTIME represents a major step forward because it learns to predict spatially resolved single-cell states. It is modeling the internal state of individual cells within their specific locations in the tissue.
That's the key.
That spatial resolution is the essential ingredient for true tumor microenvironment modeling. You need to know not just that CD8 plus T cells are present, but that they are clustered right next to the tumor margin or that they are excluded entirely.
So GigaTIME functions as a cross-modal AI translator. It takes that cheap, routinely available H&E whole slide image as its input,
Right.
And its output is a detailed, high-resolution, spatially resolved, single-cell state prediction across 21 specific protein channels. It is, in effect, generating a virtual MIF image.
That is the core technical breakthrough. It has successfully learned the nonlinear, really subtle connection points between the visual morphology in the H&E and the molecular biology, the complex cell states revealed by spatial proteomics.
It's bridging two worlds.
It is. By translating across these modalities, it makes the scarce, expensive data virtually abundant and accessible.
So for our listeners, when a model performs such a complex translation, the question is always, how reliable is the output? I mean, how do we know this virtual data isn't just a hopeful guess?
That's absolutely the right question to ask. The research team rigorously compared GigaTIME's translation performance against prior state-of-the-art methods, things like generative adversarial networks, using objective measures of image similarity and correlation. And the results show that GigaTIME was significantly more accurate and more robust than these previous AI translation approaches. The model didn't just guess. It demonstrated a statistical fidelity that makes the virtual data trustworthy enough for large-scale discovery.
And that accuracy, as always in the world of Gen AI, is directly attributable to the gargantuan scale of the training data.
Precisely. This was the first large-scale study of its kind, exploring multimodal AI for generating virtual mIF, and the paired dataset they assembled was it was truly immense.
How immense are we talking? Give us the numbers that made this model so fluent in the language of cells.
We are talking about an unprecedented training dataset drawn from the Providence Health System archives, 40 million cells in total.
40 million.
Each one with paired H&E and actual IF images covering those 21 crucial protein channels.
Wow.
Think of the AI as a student learning a complex language. When you give it 40 million paired examples, the morphological look on one side and the molecular reality on the other, it truly learns the subtle, hidden language connecting cell shape to cellular function with a level of detail that was, previously unimaginable.
That scale is what moves the translation from a proof of concept into a reliable, real-world discovery tool. So we move now from the technical breakthrough of the translator itself to the sheer magnitude of its application.
Yeah.
They built the GigaTIME model, and now they turn it loose on the archives.
Mm-hmm.
What kind of virtual population were they able to generate, and crucially, what makes this population so special?
The scope is transformative. They applied GigaTIME across the Providence real-world data system, which gave them access to this diverse cohort encompassing 14,256 cancer patients.
14,000.
And these patients spanned 51 hospitals and over 1,000 clinics within that single system, which ensures a high degree of demographic and clinical diversity.
So this is real-world evidence. Messy.
It's the definition of robust, real-world clinical evidence, often much messier but ultimately more representative than perfectly curated academic data sets.
And by applying the AI translator to all 14,000 plus patients, they generated roughly 300,000 images.
Approximately 300,000 virtual mIF images in total. What? This is the moment where the bottleneck just shifts. The significance here cannot be overstated. This scale made it the first ever population-scale study of the tumor immune microenvironment based on high-resolution spatial proteomics.
The first ever.
To get 300,000 actual mIF images, you would be looking at an investment in the billions of dollars and decades of laboratory work.
Billions?
They achieved this scale virtually for essentially the cost of computational time.
That shift from billions of dollars and decades to a computational pipeline, that's the paradigm change we're highlighting.
It is.
Instead of meticulously studying a small cohort of expensive samples, researchers now have a massive, intricate virtual landscape to explore patterns, connections, and system-level dynamics.
That's right. It moves research from constrained small-scale hypothesis testing to large-scale data mining and discovery.
So once this immense data haul is generated, how do researchers actually organize and interact with it? I mean, you can't just scroll through 300,000 images.
No, absolutely not. The visualization and organization are critical. The researchers organize this massive data haul into what they call a time spectrum.
A time spectrum.
If you look at the visualization in the paper, it presents as this complex hierarchical circular plot. You can think of it almost like a stellar map of cancer subtypes.
Okay.
It visualizes the averaged virtual mIF activation scores across all 21 protein channels at the population level.
Tell us more about that visualization. How would a researcher, say, looking for a new therapeutic target, actually use this map?
Well, they're essentially layering the clinical classification data directly onto the virtual imaging data. So the inner circle of the spectrum encodes the cancer classification, specifically the OncoTree classification, which categorized the 14,256 patients into 306 specific cancer subtypes.
306.
Mm-hmm. And the outer rings then group these activations into the 24 major cancer types.
So it's a nested navigable structure, like a zoomable map.
Exactly. Imagine a researcher focused on, say, glioblastoma multiforme. They can immediately zoom into that specific region of the time spectrum.
Okay.
They can then visually compare the immune cell activation patterns, the virtual CD8 or PD-L1 scores for that subtype against, say, the patterns of a different brain tumor subtype, or against all lung cancers.
You can spot the differences instantly.
This hierarchical structure allows for immediate visual comparison and deeper data exploration. It helps them quickly identify if a particular immune signature is common across many types, or if it's unique to a specific rare subtype.
It's the difference between studying a single patient's biopsy under a microscope and having a high-resolution interactive map of an entire species' biological characteristics.
That's a great analogy.
It allows them to understand the ecosystem, not just a single tree. This ability to instantly compare the time landscape across 306 subtypes is a staggering leap forward. The real payoff for generating this massive virtual population is, of course, the discovery phase. When you shift from data scarcity to data abundance, 300,000 virtual mIF slides, what kind of novel insights or associations were uncovered?
The volume of discovery is frankly staggering, and it highlights the power of sheer scale. The virtual population uncovered 1,234 statistically significant associations.
1,234.
Yeah, linking the virtual mIF protein activations with critical clinical attributes. And these attributes range from established genetic biomarkers to pathological staging and, most importantly, patient survival rates.
Over 1,000 validated links to just waiting to be explored. That fundamentally shifts the research methodology from confirmation to exploration. Let's dive into the biomarker associations, because that's where the immediate link to treatment planning happens.
Okay, so first, we saw important validation of existing knowledge, which is absolutely essential for trusting the virtual model.
You have to prove it can find what we already know.
Exactly. The virtual population corroborated established clinical findings that MSI, high microsatellite instability, and TMB, high tumor mutation burden, are strongly associated with increased activations of time-related protein channels, like CD138.
Okay, so it checks out.
It checks out. If the virtual data can confirm existing expensive findings, we know the underlying translation is reliable.
Now, for the novel discoveries, the things they couldn't have found before because they didn't have the scale for a pan-cancer analysis, what previously unknown associations jumped out?
Well, the immense scale-enabled pan-cancer analyses that yielded new, crucial associations between immune activation and specific genetic drivers. Specifically, GigaTIME uncovered pan-cancer links between immune activity and two biomarkers that are critical in oncology. The tumor suppressor, KMT2D, and the oncogene, KRAS.
Let's pause on that, because those aren't trivial targets.
Yeah.
KRAS in particular has long been infamous in oncology.
Exactly. KRAS mutations are present in a significant percentage of human cancers, lung, colorectal, pancreatic cancer. For decades, it was considered undruggable because of its structure.
Right.
And while recent breakthroughs have found ways to target specific KRAS variants, linking KRAS status to pan-cancer immune activation patterns is huge. It suggests that the presence of this notorious oncogene drives specific, potentially targetable changes in the surrounding time, regardless of the tissue of origin.
And KMT2D is a critical tumor suppressor.
Right. KMT2D is involved in epigenetic regulation. Its loss or mutation is often a predictor of poor prognosis in cancers like lymphoma and bladder cancer.
So finding a link is significant.
Finding a robust correlation between specific immune cell activations and the status of these foundational genetic regulators, KRAS and KMT2D, across dozens of cancer types means researchers now have a new line of attack.
How so?
Well, they can look at targeting the specific immune environment driven by that genetic mutation rather than solely focusing on the gene product itself. This just opens vast avenues for new drug combinations.
So the model wasn't just good at associating cell states with genetic markers. It was highly effective at predicting patient outcome. The paper details how this virtual population enabled sharp patient stratification for prognosis.
That's correct. Patient stratification is the ability to group patients based on expected outcomes, whether that's pathological stage or long-term survival.
Right.
Researchers have long known that certain individual immune proteins like CD3 and CD8, which mark T cells, are key prognostic indicators. GigaTIME simulated versions of these individual proteins proved to be highly effective in stratifying patients based on survival, validating the output against known clinical standards.
But the finding that truly shows the power of AI modeling is the value of the holistic view, the big picture.
Absolutely. I mean, the complexity of the time demands a complex analysis. The combined GigaTIME signature, which uses the predictive information from all 21 virtual protein channels simultaneously,
All of it one.
achieved even better patient stratification compared to using any single channel alone or even a basic combination of known markers.
So the whole is greater than the sum of its parts.
It confirms the hypothesis that the critical information lies not in one single marker, but in the nonlinear interactions of the entire cellular system. The predictive power is in the ensemble, not the soloist.
That concept of nonlinear interaction leads us right into the discussion of spatial context. The virtual data didn't just tell them what was there, it told them how it was organized.
And this is where the virtual mIF truly shone. The virtual population allowed researchers to go far beyond simple density counts and look at sophisticated spatial features. For example, the model revealed associations between virtual mIF activation density and specific geometric or organizational metrics like sharpness and entropy.
How do those spatial terms, sharpness and entropy, relate to biology?
So think of sharpness as how distinct the boundary is between the tumor and the immune infiltration. High sharpness might suggest a very clearly demarcated, excluded cold tumor margin.
A hard wall.
A hard wall. Entropy, on the other hand, describes the organizational disorder or randomness of the cells within the time. High entropy might mean immune cells are scattered chaotically, while low entropy might mean they're neatly clustered.
I see.
The model was able to link these detailed spatial metrics, how organized the cells are, directly to clinical biomarkers like APC and, again, KMT2D. This is incredibly granular biological insight that simply wasn't scalable or accessible before.
So the geometry matters.
It suggests that the geometry of the immune response is as important as the components themselves.
And this capability to study combinations and pathways seems incredibly useful for designing next-generation combination therapies.
Oh, it's essential. Most modern cancer treatments involve combinations of drugs because cancer is a combinatorial disease. But identifying the most impactful protein pairs is difficult.
Right.
GigaTIME enabled extensive combinatorial studies. The researchers used logical OR operations for pairing protein channels, for instance, looking at the presence of either CD138 or CD68, or the pairing of PDL1 and caspase-3.
And what did they find?
They found that these combined combinatorial channels demonstrated substantially improved associations with clinical biomarkers compared to analyzing either single channel in isolation.
So it lets you screen for the best drug combinations virtually.
It allows researchers to quickly screen which therapeutic combinations are most likely to correlate with a patient's genetic profile. This shifts the drug discovery timeline dramatically.
So for any breakthrough of this magnitude, especially one built on generative AI, the critical next step has to be independent external validation.
Absolutely.
They developed GigaTIME using the Providence system data, so they absolutely needed to prove the findings weren't just specific to that single institution.
Validation is non-negotiable for clinical acceptance, and the research team tackled this challenge head-on by applying GigaTIME to a massive, publicly available independent resource, the Cancer Genome Atlas, or TCGA, dataset.
The TCGA is a cornerstone of modern cancer research. It provides comprehensive genomic and clinical data for thousands of patients. How large was that validation population for GigaTIME?
They applied GigaTIME to 10,200 patients within TCGA. The goal was to study how well the virtual mIF generated by GigaTIME predicted the clinical biomarkers that were available in that distinct dataset.
So basically asking, does the AI model still work when applied to samples collected and processed by different labs under different protocols?
Exactly that, a test of generalizability.
And did these two massive virtual populations, Providence and TCGA, agree on the overall biological landscape?
They showed a remarkable concordance. When they compared the virtual protein activations across all the cancer subtypes between the Providence and the TCGA populations, they found a very strong Spearman correlation of 0.88.
0.8? That's very high.
That indicates a very high degree of statistical agreement across the two patient cohorts.
So is that level of correlation 0.88, is that sufficient for immediate clinical use, or is it more of a strong foundation for research discovery right now?
Right now, this correlation is the perfect foundation for research discovery. It provides great confidence that GigaTIME is translating H&E into mIF in a biologically robust, generalizable way.
It's learning the rules.
It confirms that the AI is learning fundamental rules about cell morphology and state, rather than just memorizing features specific to the Providence archive, for full, prospective clinical application.
Like deciding a patient's treatment based only on the virtual data.
Exactly. For that, we would want to see that number climb even higher, but for powering discovery research, 0.88 is a resounding success.
And the concordance extended beyond just the general activation scores. It applied to the actual discoveries they made.
Yes. They found a very significant overlap of the associations between the GigaTIME simulated protein activations and the clinical biomarkers across the two populations. A rigorous statistical test confirmed this high level of agreement, which corroborates many of the 12,234 original findings.
So the discoveries are real.
This is key. Validation means the discoveries are likely real, not just artifacts of the training data.
That said, there was a key difference between the two datasets that highlights the continuing and, increasing value of large, diverse, real-world data in the age of virtual patients.
That difference is crucial. While TCGA validated the model beautifully, the Providence virtual population, the real-world evidence cohort, actually yielded 33% more significant associations than the TCGA population.
33% more. Why the difference? They're both massive populations.
The difference lies in the breadth and depth of the clinical data associated with the tissue samples. TCGA is standardized and excellent for genomics, but real-world evidence systems like Providence capture the full complexity of the patient journey.
Right.
Diverse treatments, varied outcomes, and continuous clinical updates over time that aren't available in the more static, public TCGA dataset.
So the messiness is actually a feature, not a bug.
It emphasizes that while public datasets are superb for validation, large, diverse, and dynamic real-world evidence systems remain absolutely essential for driving novel clinical discovery. They capture the nuances of a much broader patient population.
So GigaTIME is operational, validated, and generating massive insights right now. But the researchers clearly view this incredible accomplishment not as an endpoint, but as a critical first step towards something much, much bigger.
This is truly a moonshot ambition. The researchers describe GigaTIME as a promising step toward the moonshot goal of developing a complete virtual patient.
Wow.
This is the ultimate aspiration of personalized medicine enabled by AI.
A full digital twin.
A high-fidelity digital twin, yes. The vision is that this virtual patient would integrate all aspects of a patient's biology, genomic data, radiological imaging, and now high-resolution spatial proteomics via GigaTIME to accurately forecast disease progression.
Yeah.
But even more powerfully, it could forecast counterfactual treatment responses.
Explain that concept of counterfactual treatment responses for us.
Counterfactual means what would happen if. The AI could predict what would happen if we applied this specific new drug or this combination to this patient's specific tumor microenvironment, even though this patient has never received it.
It's a simulation.
The model would simulate the outcome of a treatment plan without the patient ever having to undergo the risk, the time, or the expense of a trial.
That level of foresight changes the entire clinical landscape. It moves medical practice from reactive treatment based on past population averages to proactive, optimized, and personalized intervention based on a digital prediction.
And critically, to ensure this field accelerates, the research team made a commitment to open science. They didn't hoard this technology. They made the GigaTIME model publicly available on Microsoft Foundry Labs and Hugging Face.
So anyone can use it.
That open access commitment means the broader clinical research community can immediately apply these tools, generate their own virtual mIF populations from their own archives, and start building upon these discoveries today.
If we connect this back to the bigger picture, GigaTIME's most profound impact isn't just the number of associations it found, but the fact that it demonstrated the potential of multimodal AI to fundamentally scale real-world evidence generation.
For decades, the constraint was generating the high-resolution, high-cost cell state signals, the mIF data needed for analysis.
And GigaTIME converts the cheap, routinely available cell morphology data, the H&E slides, into that scarce high-resolution signal. It's performing a sort of data alchemy.
Exactly. This conversion shifts the entire bottleneck of cancer research. Now, the constraint isn't generating the data, it's analyzing the massive volume of high-fidelity virtual data we suddenly have access to.
We've gone from data scarcity to data abundance overnight.
We have. And every hypothesis that was too expensive to test is now on the table.
This fundamental change creates fascinating and immediate growth opportunities for future research efforts, which the team is already planning.
Definitely. The immediate next steps involve enhancing the model, handling more spatial modalities, increasing the number of cell state channels beyond the initial 21, and refining the resolution even further.
But the really advanced goal involves integrating GigaTIME's output into these sophisticated, cutting-edge multimodal frameworks like AlaviMed. ALVED. That name sounds like a combination of language and vision capabilities applied to medical data. How would that integrate with the GigaTIME output?
It would facilitate what is called conversational image analysis. Imagine AlaviMed acting as a massive intelligent interface between the human researcher and the 300,000 virtual images.
So it's like a search engine for the data.
It's like turning a massive data pipeline into a sophisticated, highly specific Google search bar for cancer research.
So instead of needing a PhD in biostatistics to write complex code to query the data, a pathologist could just ask a natural language question.
Precisely. Instead of running complex computational pipelines, a researcher could literally talk to the data.
What would that sound like?
They could ask the AI, in plain English, show me all the lung cancer subtypes in patients who showed poor survival rates, where the spatial density of CD8 cells is high and the KMT2D tumor suppressor is mutated.
And the system would just find it.
The system then processes that conversational query across the entire virtual population and returns the highly specific subset of images and data points.
That accelerates the cycle of discovery exponentially. It moves therapeutic discovery at the speed of conversation, allowing medical professionals to interrogate the data using their clinical intuition rather than being limited by coding knowledge.
It is about reducing the time from clinical question to actionable insight from weeks or months down to minutes.
So what does this all mean? The ability to generate 300,000 virtual mIFs of patient tumors, and potentially millions more down the line, means researchers are no longer constrained by the $5,000 cost of a single tissue sample analysis. This is about unlocking knowledge that was previously inaccessible simply due to budget constraints, freeing researchers from that economic prison.
That's right.
This capability allows researchers to test thousands of complex biological hypotheses simultaneously across vast patient populations instantly. The method of scientific inquiry moves from slow, expensive, specific hypothesis confirmation to rapid, comprehensive data exploration.
It fundamentally changes the cost structure and the time horizon of hypothesis testing. It makes advanced spatial biology accessible to virtually any researcher with access to H&E slides.
Which raises an important question for you, the listener, to mull over. When complex, critical biological data about life, death, and disease response becomes virtually limitless and easily accessible, how fundamentally does that change the speed and methodology of therapeutic discovery? We may be witnessing the true beginning of accelerated precision medicine, where the only limit is the researcher's imagination.